Welcome back. This is day 9 of genetics, and today we're going to be talking about how can biotechnology increase our quality of life. So again, today we're going to be talking more about genetic engineering, as well as the pros and cons associated with it. But before we get to that, you guys have got 2 do now questions, so go ahead and pause this video, answer questions 1 and 2, and press play when you're ready to review them. Number 1, individual cells can be isolated from a mature plant and grown with special mixtures of growth hormones to produce a number of genetically identical plants. This process is known as what? Well, I can think of a process that would produce genetically identical copies, right? That process is known as cloning. I really wish I could clone myself. I need someone to run my errands for me. But alas, here we are. Number 2, to produce large tomatoes that are resistant to cracking and splitting, some seed companies use the pollen from one variety of tomato plant to fertilize a different variety of tomato plant. This process is an example of what? Well, in this example, we are choosing which 2 species or which 2 varieties of plants are mating together in order to produce a new variety with the best qualities. This process is known as aselective breeding. All right. Which leads us to, what exactly is genetic engineering? So you guys do need to fill in these blanks on your paper. Genetic engineering is the direct modification of an organism's genome. And we learned today that this word genome is referring to the list of specific traits or genes stored in the DNA. So it's basically just the collection of genes that you have. Now, changing the genome enables engineers to give desirable properties to different organisms. So when we see this phrase desirable properties, this just means good traits, whatever traits that the engineer wants that organism to have. Now, finally, organisms created by genetic engineering are called genetically modified organisms, which we sometimes abbreviate GMOs. Now, you may have actually seen this phrase GMOs at the grocery store before, because if any product is genetically modified, it is required to say this is a GMO product on the label. And oftentimes, labels will even say this is a non-GMO or non-genetically modified. So today we're going to be talking about the pros and cons associated with each of those techniques. But let's look at what this process actually looks like. So today in class, we talked about CRISPR, but this is not the only form of genetic engineering we have. In fact, we've actually been doing this for quite some time now. So humans are learning how to manipulate DNA, and we do this by using restriction enzymes to cut open a bacterial plasmid. Remember, our restriction enzymes are those molecular scissors, right? So this right here is representing a restriction enzyme that cuts the DNA, and we're going to use those restriction enzymes to cut out a human gene. So this is what we see right here. The human gene we're looking at in this example is an insulin gene. So then once we remove that insulin gene, we can insert the human gene into a bacterial plasmid. So this circular structure right here, that in blue, is our bacterial plasmid. And a plasmid is just a word that means bacterial DNA. Okay. So we can actually insert this human insulin gene into bacteria, but I want to pose the question, why are we using bacteria? What do we know about bacteria that might be kind of helpful for us? I don't know. I'm going to leave that open-ended. We'll see if we can figure it out in just a second. So let's look at the process by which, hold on, let me back up. I'm going to explain each of the steps involved with this process. We're going to break it down so we can see how it starts and what the goal at the end is. This bacterial plasmid. Again, this is just a word that means bacterial DNA. Right? We're going to use our restriction enzyme to cut open that plasmid. So in this example, we're not using scissors, but I'll draw us some just to show you my beautiful artwork. Now, once we cut open that bacterial plasmid, we also need to do the same thing with our restriction enzyme to cut open or cut out the specific gene that we're trying to insert into our bacteria. So in this example, we can say that that gene of interest is an insulin gene, specifically a human insulin gene. So we're going to use our restriction enzymes, we're going to use our scissors, and we're going to cut out that insulin gene. But then what's our next step? We want to combine those two things, right? But I don't think they're just going to stick together on their own. So we actually have to bring in the help. Oh, my animations are out of order. That's okay. We're actually going to bring in the help of another enzyme called DNA ligase. Right? DNA ligase is an enzyme that basically acts as glue to seal together this human DNA with the bacterial DNA. Right? So this is glue that seals together the different DNA. Now, once we seal our human trait or our human gene into our bacterial DNA, we're then going to call this result recombinant DNA. Now, when I see this word recombinant, it kind of looks like the word combined, right? And that is because we are combining the different types of DNA. So in this example, we're combining human DNA. I'll do that in green. Great, this is our insulin gene. So this is now human DNA plus bacterial DNA. And that means this is now recombinant DNA, right? We've combined those different types of DNA, right? Now, we're then going to insert that recombinant DNA back into the bacteria. Now, earlier I asked you guys, why do we want to use bacteria in this process? Why are we trying to stick a human insulin gene into bacteria? Well, since the bacteria now has this gene, that bacteria can now produce insulin. It now has the instructions to produce insulin. And why is that helpful? Well, think about how bacteria reproduce, right? They actually reproduce asexually, which means two things. They reproduce fast, and all of their offspring are identical, which means that now all of these resulting bacteria can do what? They can all produce insulin as well, right? These can all produce human insulin. So this is how we have actually created the mass production of insulin. So if you have anyone in your family who takes insulin shots for diabetes, hold on, mass production. This is most likely where they get their insulin supply from. This is a super cheap and a super fast and a super easy way to produce insulin by having bacteria do the hard work for us, right? So we cut out the human gene that codes for insulin. We place it into a bacterial plasmid. We then put that plasmid back into the bacteria. It starts reproducing, and all of its offspring can now produce insulin. So then scientists can bottle it up and sell it to anyone who needs it, right? So now this same technology could be applied to many different scenarios. And it makes you think, could Spider-Man be real? Because, right, we could cut out the gene that codes for silk production in a spider, and we could put it in a human. So could he be real? Well, possibly, but there are a lot of ethical concerns surrounding this. So we haven't used a lot of this technology on humans just yet, but we have used it on goats. So we've actually created web-producing goats. We took these spider genes and actually inserted them to goats. So we can now have goats that produce silk in their goat milk. We've also done this with a lot of other organisms as well. We've used the same technique to create fast-growing salmon. We took genes from other fish that cause this salmon to continuously produce growth hormones, which means it's going to get bigger faster. And this is really helpful for fishermen, right, who want to have a greater turnaround and make more money faster, because those fish grow faster, which means they can sell that meat quicker. We've also used this to produce less smelly cows. So we've modified the bacteria responsible for methane production in cattle, which results in 25% less flatulent cows. So this is good because it means that there's less CO2 being produced by their farts, but it's also good for the farmers who have to stick around these cows all day because they're not quite as smelly. Now, this history of GMO development is actually pretty recent technology. So in 1973, we first created the first genetically modified bacteria. So that's really the first time this technology was used. But then the next year we turned around and we actually created genetically modified mice. So we started upping our game pretty fast. In 1982, we had the first commercial development of GMOs. And this is when we actually started using that technique I just showed you to produce insulin by using bacteria. So that is when we first started using that insulin technology. In 94, we began to sell genetically modified foods, those GMO products you might see at the store. And then in 2003, we actually began to sell GMOs as pets. You may have seen these glowfish at the pet store before. I know that I'm always really tempted to buy one whenever I see them. I haven't just yet though, but essentially these glowfish were produced by isolating the bioluminescence gene that's found in a lot of marine animals. So jellyfish and some bacteria that you might see in the sand or the water at night. And they actually inserted that gene into fish that allows them to glow. So if anyone has a glowfish, please send me a picture. I'm really jealous and I think I might go impulsively buy a glowfish. All right, now I do want to ask you guys a question, and that is, how do you think that genetic engineering, or how might genetic engineering modify plants to solve everyday problems? So think about problems we face, such as world hunger, weather problems, insecticide pollution, and think about how could genetic engineering fix these problems? All right, pause this video, answer that question, let me know what you think. Now, there is no one right answer for this question, but I am about to show you some of the pros and cons associated with this. So you do need to fill these blanks in on your paper. Here are some reasons that we could or might want to genetically modify plants. These are some good or some pros. So we can genetically modify crops to be insect resistant or herbicide resistant or drought and freeze resistant, even disease resistant, right? These are really helpful things to have. We could actually genetically modify them to have a higher yield, which means produce more. We could genetically modify them to grow faster. We can also genetically modify them to have improved nutrition to help us out more, as well as have a longer shelf life so they last longer. These are all reasons why genetic modified crops are a good thing that could really help us out in solving some of those issues that we face. But there's also some concerns surrounding GMOs and some reasons we might not want to do this. And that is because this is pretty recent technology, they might pose a risk to human health. They might be unsafe to eat. We don't really have long-term research on how this affects us in the long run. The same thing, they might pose some sort of harm to the environment and wildlife. Again, we don't have long-term research to know what effect this is going to have down the road. They might lead to increased pesticide and herbicide use as those plants evolve. And this also might pose a problem to farmers' health as they are around those increased pesticides and herbicide use. Seed and pollen drift is also another risk. There are some concerns about whether pollen from a farm that uses genetically modified crops will blow over from the wind to a farm that doesn't use genetically modified crops. What happens if those plants cross over? What if those plants, through sexual reproduction, create some sort of hybrid that is, I don't know, I don't want to make any guesses here, but there are just concerns about how that will affect the plants as well as the environment. There's also the possibility that we could create herbicide-resistant superweeds. So as plants evolve to the herbicides that we use, those plants get stronger. They learn how to adapt, right? Through mutations, they might become resistant to those herbicides we use, which means we might have to use stronger and more just toxic herbicides, which is just not good for the environment. And now this also opens the door to, what about genetic engineering in humans? So this is something that was addressed in that documentary we watched today. Is this ethical? If it is, where do we draw the line between what is and what isn't? There's just a lot of questions that we don't really have all the answers to, and there's a lot of differing opinions about what is and what isn't right. So nearly 50 countries around the world, including Australia, Japan, and all of the countries in the European Union, have enacted significant restrictions or full bans on the production and sale of genetically modified organism food products, And 64 countries now have GMO labeling requirements. So this is why typically in the store you will see that food products are either labeled as GMO or non-GMO. This is because of the regulations that have been enacted. Right, so I've got two more things for you guys before I let you go. And that is number one, we've got a question. The diagram below illustrates some key steps of a procedure in one area of biotechnology. This is that same insulin example I gave you guys earlier. So we cut out the human gene for insulin. We cut the bacterial DNA, right? This is known as a plasmid. And remember, what enzyme did we use to cut out those things? We used our friends, the restriction enzymes. Always cutting our DNA. And then we actually used a new enzyme, which I told you about today, to glue together those two pieces, right? That was called DNA ligase. So it glues or seals the DNA. And then we insert that bacteria, that recombinant DNA, into our bacterial cell. But then those cells are going to start to divide and lead to X. So what does that letter X most likely represent? Does it represent bacterial cells that are unable to synthesize insulin? Does it represent human cells that are able to synthesize antibodies? Does it represent bacterial cells that are able to synthesize insulin? Or human cells that are unable to resist antibiotics? Well, the answer here was C. It represents bacterial cells that are able to synthesize insulin, right? They now have that insulin gene, which means they're able to produce insulin. Now, 4 vocabulary words for you guys that you have heard before. So what is the circular DNA structure used by bacteria? That is that word we described as a plasmid. So a plasmid is bacterial DNA. All right, what is an enzyme that cuts DNA whenever specific base sequences are present? We've heard this one before. These are our molecular scissors called restriction enzymes. All right, next up, we've got DNA in which a section has been removed and replaced or recombined with a new sequence. Remember, that is when we combine our different types of DNA. So this is known as recombinant DNA. So we saw like when we combined our human DNA with our bacterial DNA, that's recombinant DNA. All right, and finally, an enzyme that combines DNA bases. This was our glue that glues those different those different segments together, which is known as DNA ligase. All right, the last two questions I do want you guys to answer on your own using what we learned today in class. All right, other than that, all you need to do is fill out your exit ticket, submit these notes, and have a wonderful weekend. I'll see you guys on Monday.